There are many ways to determine the three-dimensional structure of a molecule to near atomic resolution. But the dominant method, and often the most precise, is X-ray crystallography. X-ray crystallography has helped us to understand the relationship between a molecule's structure and its function. This includes small molecules like drugs, metal complexes such as porous metal organic frameworks, and even large biological macromolecules, which are proteins, DNA, and RNA. To date, crystallography has contributed to 29 Nobel Prizes and 48 laureates. The crux of crystallography, however, depends on growing single crystals of sufficient size and quality to diffract X-rays. And this can be a real challenge, because very often crystals grow stuck together, can be only a few microns in size, or contain defects. This is where the Proxima II beamline comes into play with its intense and microfocused X-ray beam, which can select specific zones of a larger imperfect crystals to record clean X-ray diffraction patterns. Because crystallizing molecules is so fickle, scientists often need to screen hundreds of crystals to obtain the best possible X-ray diffraction data, which will lead to their crystal structure.
The X-ray source on Proxima IIa is a powerful in-vacuum undulator, which provides an intense and highly collimated X-ray beam. The optical design is simple and elegant. The monochromator is a cryo-cooled silicon 111 channel cut crystal that selects the X-ray energy from 6 to 18 kiloelectron volts, which is suitable for most SAD and MAD experiments. The beam is focused down to 10 times 5 microns by a pair of adaptive X-ray mirrors. Together, the source and optics deliver an X-ray flux in excess of 10 to 12 photons per second. The Proxima IIa end station is optimized for microcrystallography experiment and consists of multi-axis goniometer, a large sample change robot, a cryostream, and a large fast X-ray area detector. The goniometer is equipped with a multiple high-performance rotation axis, sub-micron resolution centering stages, and an on-axis microscope, which allows us to see the sample as the X-ray beam does. The robotic sample changer automatically transfers samples mounted on spine pins from the dewar to the goniometer. The dewar can hold 9 unipucks for a total of 144 spine pins. The time to mount and unmount between two samples is less than 30 seconds. The X-ray area detector is an Eiger 9M, which collects X-ray diffraction data at 238 frames per second. A full 360 degrees of X-ray data is typically collected in less than 16 seconds. Each X-ray diffraction image consists of over 10 million pixels, and a guided user interface called MXCube allows the scientists to set up the crystallographic experiment. This includes loading and unloading samples with the robot, centering the sample, setting the goniometer angles, the detector distance, the X-ray energy, and naming the data files.
To help scientists quickly screen through and collect data from hundreds of crystals, Proxima IIa has implemented several automated features and functionalities. For example, we have developed a machine learning algorithm that segments out sample optical images, automatically aligns the sample with the optical center of the camera and the X-ray beam. It is fast and efficient and measures sample geometry in the process. The sample zone of a crystal can be aligned with beam either optically or with X-rays. Optical alignment is done by clicking at a particular zone of the sample at multiple orientations. Due to refractive effects, a layer of mother liquor can distort visual position of the crystal, making its optical alignment difficult. For such cases, we have developed a method that accounts for such refractive effects explicitly and allowed to model sample environment in the process. X-ray centering is particularly useful for very small crystals, which can easily rotate out of the microfocused X-ray beam. This functionality scans across the sample at multiple orientations, and reconstructs the diffracting volume shape in the process, using that information then to perfectly align it with X-ray beam. Some crystals are transparent and almost invisible in the mother liquor. Fast grid or mesh scans help to locate the crystal and also help to find the sweet spots, the parts of the crystal that diffract X-rays the best.
Proxima IIa has developed its own crystallization plate screener, known as the Kribbler, which can be adapted to any SBS format plate. This device has drawn the attention of scientists working on protein crystals that degrade during the cryo-freezing step, because it allows users to collect X-ray diffraction data directly in the crystallization plate, under ambient conditions, and forgoes any damage to the fragile crystals during the fishing and the freezing steps.
Since Proxima II opened to the scientific community in 2013, it has hosted a large number of users from the international scientific community. These include not only scientists from France and Europe, but also from the USA, Brazil, Argentina, Saudi Arabia, and India. Many of the results from Proxima II have appeared in high-impact journals, such as Science and Nature, which cover a wide range of topics including virology, microbiology, and cancer. Proxima II also regularly hosts industrial users who are developing new medicines and vaccines. Finally, as part of our user program, Proxima II offers practical training sessions on the beamline so that the users develop confidence when they are collecting X-ray diffraction data from their precious crystals.